So RNA and protein synthesis. This was originally supposed to be part of the DNA unit, but because of time and the presence of finals coming up, I decided to cut it short, do a very, very small segment on DNA, and then just push the rest of this to the next semester, which actually turned out to be great timing with the virus and the vaccine and all that stuff. I'm super hyped about being able to use what we're learning and use it to understand some really relevant stuff that's happening right now. All right, so also because this is supposed to be part of the last unit, some stuff should feel familiar, sound similar, so it won't be as daunting a task. There are some similar processes between RNA replication, or I guess transcription, and DNA replication. Just a couple little small differences. All right, so getting started, our genes determine our traits. Genes are that code of DNA that we have in all of our cells. So how does this work? Why is this important? So our gene, or that code of DNA, is found on our chromosomes. Chromosomes are made up of that DNA. They twist and wind to form the chromatin, which twists and winds to form our chromosomes. We don't want the whole strand of DNA information if we're just making one protein. So let's say I have my skin cell and my finger here. Inside my skin cell, in the nucleus, we have all of the DNA for everything in our whole body. Obviously, this little skin cell is not going to display traits or aspects of my being that's not useful to it. My skin cell is not going to need functions that are the same as like a liver cell or a lung cell. That's why it's important that we have this RNA and protein synthesis. Traits are things like our hair color, our eye color, our height, things like that. Again, all of our cells have nuclei. The nuclei contains all of the DNA for our entire being inside of it. When we have our individual cells, they produce proteins that are important to the function of that cell. Again, my finger's skin cell is not going to need to know how to process oxygen like a lung cell. Each cell in our body, or each type of cell in our body, needs different things to carry out its specific function. So our genes, in our example on the right here, we have a chromosome. I'm just choosing a random spot on this chromosome. It happens to be on the end of the right arm here. Let's just say that's the location of this protein. I'm using melanin as an example of a protein. So melanin, the code for that protein, is found in this spot on this chromosome. I just chose something randomly. I don't actually know where that sequence is. And that melanin is a protein that displays a trait of skin color. Some people have a different amount of melanin, giving them a different shade or color of skin. So our genes produce proteins, and those proteins give us traits. Here's just another way of saying it, how DNA codes for things. So on the left, we have our DNA. DNA gives codes for proteins. Proteins tell our cells what to do, and those cells make us up. Now we're made of cells.
How do the cells know which instructions to follow, since they don't need all, like how does that happen?
We're going to get to that. That's an excellent question. You're just
Sorry
on ahead of me. No worries. I appreciate it.
That was
you're asking the right questions. So as a quick review, DNA is made of 4 letters, as we know: A, G, T, and C, adenine, guanine, thymine, and cytosine. And those 4 letters, again, they make up the code for everything in our body. Kind of crazy that 4 little things can tell our entire body what to do specifically depending on its needs and its functions. Well, looking at a different polymer, protein.
Mr. Aleph, can you go back for like 10 seconds? I missed something.
Absolutely. So thinking about, sure thing, macromolecules, unit 1, really, really long time ago. We have our nucleic acids. DNA is a nucleic acid. We have our proteins. Proteins are a macromolecule. And so each of those macromolecules are made up of monomers. DNA, those monomers are nucleotides, the A, G, T's, and C's. Proteins, their monomers are these amino acids. So just like how DNA has 4 different types of nucleotides or monomers, proteins have 20 different types of monomers or amino acids. So depending on the order of those amino acids, just like in DNA, we can get a different code for something different. So let's say we have a certain order of amino acids that'll give us one protein. But if we take those same amino acids and we mix up the order, we're going to get a code for a different protein. So just like DNA, how we can use the same letters but mix them up and get different things, applies also to proteins and amino acids. This means there's just an unimaginable amount of combinations to make all of these different things that we need in our bodies. I don't even know where to start counting all of the different proteins that help our body function and work. And it all comes from those 4 letters on DNA, and those 4 letters make up code for those amino acids, which make our proteins, which make our bodies work. Lots and lots of crazy, insane amounts of information. All right, we're gonna pause. We're gonna go over this important piece of vocab, dogma. A dogma is a principle or a set of rules that are incontrovertibly true. They're facts, indisputable. We know them to be true. No amount of evidence that we can find disputes these things. Something like for physics, gravity, super duper important. We know it exists everywhere, off of Earth, on Earth, other planets. It's a constant. It's always there. Just a fact. For biology, we have a dogma about proteins and how life works. DNA, which all of our cells have, make RNA, and that RNA then makes proteins. So it's just this little path, DNA to RNA to proteins. Obviously, there are steps that make that happen, which we're going to talk about. But the central dogma of biology, most important thing that runs bio, DNA makes RNA, RNA makes protein. So why do we even have RNA? Why don't we just have DNA that makes proteins? A couple of reasons. The first being an evolutionary advantage. So let's think about DNA. DNA is really, really important because it's the key, it's the code, it's the instructions for every bit of our body. Again, that skin cell on my finger has the code for brain cells. It has the code for muscle cells. Even though it doesn't need it, it still has that information. DNA is super important. If that gets messed with, mutated, damaged, broken in any way, that messes things up. It'll start that replication process that we talked about last unit, and things are going to be altered, which could potentially result in harming the organism. So the RNA fixes that problem to a certain extent. So we will have this code for protein found somewhere on a strand of DNA. Don't know which chromosome, don't know which section. I can't tell you right off the bat. I'm not some sort of research scientist who focuses on a special protein and all that stuff. But the code for a protein, let's say ligase, we've heard that protein name before. It's found on this chromosome, let's just say chromosome 10, in this one particular section. If we can only read that one section one time to make that protein, that's not very efficient. But also, if we were to say, take that segment and move it out of the nucleus to make that protein, that DNA becomes at risk. The whole purpose of that nucleus, if you remember from our organelles, main focus here, the nucleus protects that DNA. It keeps it safe because it's so important to our body, to all of those cells making things work and function. So if that were to leave the nucleus, it could get damaged, and then we'd be in some trouble. So the RNA takes some of that risk. It's making a copy of that section that's that protein. Ooh, this is This is the key. This is the code sequence, this little fragment that'll make ligase. Then I can take a copy of that information and move it out of the nucleus to make that protein. But if we take that DNA and move it out of the nucleus, it's at risk to being harmed. So by keeping the DNA in the nucleus, we keep it safe. The RNA, if that gets damaged, not nearly as big a deal. So number one, that is the evolutionary advantage. We're keeping that original important OG code safe inside the nucleus. Now we're going to go and talk about the efficiency. RNA increases the rate of protein synthesis. So we can make these copies of that sequence for, what'd I say? Ligase. So I make a copy of that and I leave the nucleus to go to a ribosome to make that protein. As soon as that code gets copied, a new piece of RNA can get made. And then we can have 10 strands of RNA to make that one protein. Instead of taking turns, oh, we're reading, we made that protein. Oh, we're reading the DNA, we made that protein. Now we can go RNA, 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 all of these copies. We can make all of these proteins super fast, super efficient. So that's another advantage to having RNA. All right, getting into the actual process here. We're going to start with the first half, transcription. Tomorrow we'll follow up with the translation. So transcription is taking the language of DNA, our main code here, and copying it, making a copy in RNA language. Very similar, only a slight difference.
If it's the
so let's take a look at RNA itself. Go ahead.
Transcription and not translation.
So transcription is more about making a copy.
Oh yeah, I guess.
Translation is what we're going to do in the second half of our protein synthesis. And that's actually translating that code into a protein.
Okay. Okay, I'll just remember it like that. Hopefully.
Good question. So we're transcribing or just making a copy at this point. So looking at our molecule of RNA here. Differences between RNA and DNA. First up, that sugar. One is called RNA, one is called DNA. That R or D tells us which one it is based on the sugar. DNA is deoxyribose, RNA is ribose. Na stands for nucleic acid, both of them are that. So the D and the R just really tell us what type of sugar is found in that backbone. The second difference comes with those nitrogen bases. Looking at our DNA, we have A, C's, T's, and G's. Great. We feel super comfortable with that. However, RNA does not have thymine. It doesn't have that nucleotide. Instead, it has a different one called uracil. We're going to look at a better diagram in a few slides about that pairing process. But whenever we have an RNA polymerase that makes the mRNA strand, whenever it comes upon an A and it goes, oh, I need to pair that. I need to match it with a T. Oh, wait, I don't have a T. It's going to reach into its little pocket, add on a uracil, and keep going. So instead of thymine, it just pairs uracil with that adenine instead. The last difference here, if you look at our diagram, DNA is on the far right. It's got two strands that are connected by their nitrogenous bases. Double ladder, still twisted, but it's got those nitrogenous bases bonded together. RNA, on the other hand, is just half of that. It's one strand, still has the sugar and phosphate backbone, different sugar, but it has those nitrogenous bases kind of hanging off the side, ready to get paired. This will become important when we go to the second part of translation. There are a lot of different types of RNA. mRNA, tRNA, rRNA, siRNA, some that I don't even know about. But for my class, we're just going to focus on two, mRNA and tRNA. So for transcription, mRNA comes into play here. The M stands for messenger. All this RNA is doing is transporting a message of that code. Here's what the DNA says. We're moving it over to the ribosome. That's its job. But in order to get that copy, we need an enzyme, RNA polymerase. Sounds super familiar. In DNA replication, we had DNA polymerase. They do almost exactly the same thing. They unwind the DNA strand. We don't have a helicase in this instance. This RNA polymerase just kind of does that because it knows that there's only a specific section that it needs to copy. It unwinds that little section, matches those base pairs, creates an mRNA strand. Just like in DNA synthesis, we're going in the 5-to-3 direction. We can only add nucleotides on that 3 end. So the orientation of the strands matters again. One will be 5', one end will be 3'. So looking at our DNA strand, if you remember, there's only a section of DNA that we want for each protein. We don't want the whole strand. We don't need all of that information. We just need the little bit that tells us how to make that one protein. Let's say like it's ATP synthase. We don't need to know how to make primase. We don't need to know how to make ligase. We just need to know how to make that one protein. And to signal where that protein starts, where that code for that particular enzyme or whatever we're making starts, we have a promoter region. It's also called a TATA box. There's like a little bit, a bunch of T's and A's on that DNA. And it's a signal. It's a lot like the primase in DNA replication. It goes, hey, over here, you want to make that enzyme? Come over here, start here. You start here, you're not going to miss anything. Over here, start here. It's a site where the RNA polymerase goes, oh, hey, there's a TATA box over there. Is that the protein I want? It is. Great. I'm going to bond, or I'm going to bind there to start unwinding and making a copy. So it's just more of an indicator of, hey, this is where that protein starts, so you don't miss any of that information. Enhancer region, oops. It's just another type or another way of saying promoter region. It's the binding site that's upstream of that gene. So again, the TATA box is like, hey, we're not actually the code for the protein, but this is a signal of where to start. All right, so what's happening at the beginning of transcription? Again, we got our transcription factors like promoter regions, that TATA box, and that triggers, it indicates, it signals where that RNA polymerase needs to bind to the DNA to start making a copy. If you remember cell communication, unit 4, I believe, we get the signal, hey, start here, hey, over here. We're gonna start over here if you wanna do this protein so you don't miss anything. The enzyme receives that signal, goes through that transduction pathway, and the response will be to bind and begin transcription. All right, here we go. Here's the diagram I like a lot. So down on the bottom, that is our DNA strand. It's got its A's, C's, T's, and G's. It's been unwound a little bit by RNA polymerase so it can read that code and make a copy. It's transcribing it. And so just like normal, C's and G's, hey, they pair with each other. It comes across a T, a thymine. Oh, great, I have some A's. So the way I like to imagine it is this RNA polymerase has like a bucket or a basket or a pocket, a bin of all of these nucleic acids. It's A's, C's, G's, and U's because RNA has uracil instead of thymine. And as it's going along, it's just going, oop, that one matches there. Oh, that one matches there. So it's like a reverse shopping cart dance. Now, when it gets to that thymine, great, I have some adenines, pair that there. But when it gets to an adenine, it doesn't have any thymines. So it's going to root around in its bin, pull out a uracil, boom, pair that there. So very, very similar to DNA replication, still in that 5-to-3 direction, but we're just swapping out that thymine with a uracil. All right. So now we've made a copy of that sequence, that little bit of the DNA code that we want to make this protein. Well, there's some junk in here. The entire code may not be the stuff that we need. There may be some stuff that we don't need. Could be, oh, well, this little bit right here is for a different protein, or this little bit, it just doesn't mean anything. It's just a random bunch of A's, T's, and C's, and G's. I don't know. What is this bit? We don't care. It's not important to the actual coding of the protein. And we can label these things we want and things we don't want. The exons are the things that we want. They're the real sequence of the gene that will help make this protein happen. Introns are the junk. We don't want that. That's the BS that's stuck in there that doesn't mean anything. The mnemonic device I use is the exons are going to be used to express that thing, that protein, that trait, whatever we need. The introns are in between the stuff we want. They're the junk. They can be taken out. They're in between the important things. So what does this look like? Why would we have all this junk? What happens to it? Well, our RNA goes through something called splicing. The junk has to leave before we can make the protein. If our ribosome reads the code and it starts writing something into our protein that it doesn't need, our protein's going to be useless. It's not going to work. So we have to get rid of the junk. And it does this by splicing. It'll get rid of the introns, squish together all the extrons, so we have one continuous code of just the stuff we need. 5th period yesterday, somebody asked, well, how does that work? How does it know what it needs and what it doesn't need? That's more AP Bio college stuff. It's not necessary that you know how that works for my class. But if you are interested, I think it's fascinating. If you go into the PowerPoint, I've added a little link here to a slide that has a great diagram on how that happens with a different enzyme called spliceosome. Spliceosome splices that mRNA. I think it's pretty cool, but again, you don't need to know it, just there if you're interested. All right, we're coming up on the end here of today's lecture. So we made a copy, we transcribed it. Our introns are gone. We're left with only the stuff we need to make this protein. We're ready to leave the nucleus because we can't make the proteins until we get to the ribosomes. We know this from the jobs of our organelles. But in order to get from the nucleus to the ribosomes, we have to travel through that cytoplasm, that jelly-like gooey stuff that holds the cell together. All the organelles need to stay and hang out in there. It's in charge of stabilizing, essentially. But there's space. Ribosomes are not directly attached to that nucleus, so they gotta travel. Well, part of the cytoplasm job is to figure out things that are good for the cell and things that are not good for the cell, things that are potentially harmful. Bacteria can sometimes get in there, foreign objects, whatever. It can send those lysosomes on there, go, hey, they're trying to hurt our cell, get them, and then it'll kill it. But we don't want that. Our mRNA is super important. We need it to make our proteins to make our cell function. So in order to protect this brand new code that we just copied, we need to tack a couple things on it to signal to the cytoplasm that, hey, we're cool, bro. We're just gonna pass on through, keep doing our job, make some proteins to make us happy. So some of the things that we need on the 5' end, again, the orientation of this molecule is important. On the 5' end, we're going to add something called a GTP cap. Just a little thing tacked on there, part of the signaling of, hey, we're cool. Without it, cytoplasm may identify it as not cool. This thing is trying to hurt us. On the 3' end, again, we usually go from 5' to 3', so we call it a tail. That's at the end of the molecule. We have what's called a poly-A tail. Poly as in many, A stands for adenine. It's just a ton of adenines, one after the other, after the other, after the other, just a long chain of adenines together, 50 to 250 of them. Now that our important sequence is bookended, got important things on either side, the signals to the cytoplasm, we're cool, we're just passing through, we want to make proteins, don't attack us, and it can be on its merry way, leaving the nucleus on its way to the ribosomes. Fun fact, the longer that poly-A tail, the longer that mRNA can stick around and be viable to produce more protein. Meaning, our mRNA, maybe with a shorter tail, can only produce like 5 proteins from it, and then it starts getting damaged and it's no good and we'll have to replace it. But one with like, I don't know, 200 adenines on the end, it's gonna last longer in the process. We can make more proteins from that one strand of mRNA. So the longer the tail, the longer it'll stick around. Just a fun fact.